Hi, everyone. Thank you for inviting me to speak. I'm in the Department of Mathematics at Western University, not to be confused with Northwestern University. We're north of that, as a matter of fact. So today I'm going to be talking about quantitative trait heritability. I'm just going to assume that you guys don't know what that is very briefly, just to get everyone sort of on the same boat. So basically, this is a way to measure the variability in a trait in a population. So VG here is the genetic variance, actually the additive genetic variance in a trait. So that is the difference, the measure of the variability between individuals in a trait attributable to genetic causes. VE is the environmental variance. So that's the variability between individuals attributable to environmental differences that they've experienced. And so heritability is just a nice way to kind of normalize the genetic component of that. So this is a very important quantity in quantitative genetics. I'm not really going to nerd out too much about it. For today's purposes, it's just a normalized measure of genetic diversity. And so from that perspective, you can sort of see what I'm about to talk about as falling within the great tradition in math bio of trying to make sense of diversity in biology in all sorts of contexts. I think quantitative trait heritability is actually a particularly interesting case study for that. And in this project, I think I've found some interesting new math stuff as well that hopefully you guys will find interesting as well. Okay, so the basic sort of thing that got people really interested in heritability from a diversity perspective is these types of figures. So this is a classic figure showing cumulative distribution functions for heritability. This is a really old figure, as you can probably tell. And the point here is that heritability tends to be really high. So in these letters here are just like different trait groups that they've done studies on. And so it's rare to see a heritability of below 10%. Typically, things are pushing up towards even like 50%. And this applies across lots of different species, lots of different traits, right? And this implies that there's a ton of genetic variation just sitting around in populations. And these are typically eukaryotic organisms, so they're not sort of cheating by having monstrous population sizes like microbes. So the question is why? And I'm happy to hear many of you are population genetics people. So our like go-to sort of explanation for a lot of things in population genetics is a model called mutation selection balance. And it's a very simple idea. Basically, we just say selection's not doing anything fancy. It's trying to pick the best genotypes. That's going to eliminate variation. Maybe drift is there. Genetic drift is in there as well, also eliminating variation. But there's always these new mutations coming along that are replenishing variation. And obviously, if you have enough of that, you can get any heritability value you like, right? You can get as much genetic variation as you like that. And so this explanation is very appealing because, as Nick was explaining, we think that a lot of quantitative traits have, they involve a lot of genes, right? So there's this enormous big target for mutations to hit and generate a bunch of variation. Now, being a mathematical biologist, that's obviously not acceptable to us. We don't care about, is it conceptually plausible? We want models and numbers, right? And that's where things start to fall apart a little bit. So I'm just going to briefly run through sort of the classic mutation selection balance model that's been used. Apologies if this is familiar to a lot of you. So unlike population genetics models, we aren't allowed to assume selection coefficients in quantitative genetics. We just assume a fitness function on a quantitative trait. So this red curve here is a Gaussian function, basically, is typically what's assumed. Its width is a parameter of the model. That's what its variance is a parameter of the model. And the blue curve is meant to be sort of the distribution of traits in the population, right? So the population is hanging out, trying to be as close to the peak of this fitness function where their fitness is highest. And they're both sort of shifted towards the peak and sort of squished to be right as close to the optimum as possible by selection. So this is called a stabilizing selection model. And if you add to this, basically a bunch of simplifications. So just make your life as simple as possible. So forget about linkage disequilibrium, forget about drift. We just have a fixed number of loci contributing to the traits. They're all additive. Nick was talking a little bit about that. They all have the same mutation rates. Gaussian fitness function. The fitness function stands still. The population is just hanging out there. So basically, just make everything as simple as possible. You get a very famous model. It's called the Ladder-Balmer model. And this is, if it was an ice cream flavor, this would be the vanilla version of mutation selection balance. And you can solve this analytically quite easily. You get this nice prediction that the genetic variance is the simple product of a few of the parameters. So there's this term L mu there. So remember, L is the number of loci, mu is the mutation rate. So that's sort of the total influx of mutations. Makes total sense that genetic variance should scale with the total influx of mutations. And then there's VS here, which is the variance of the fitness function. Obviously, if you have a wider fitness function, it permits more variance in the population. So that's all nice and intuitive. And this is, for those of you who are a little bit more familiar with quantitative genetics, you get the same result if you sort of go the other route and do the house of cards approximation. So this is quite a widely used formula. Okay, so let's put some numbers to this. So I'm going to pick on Drosophila a little bit here. So that's a recently measured mutation rate in Drosophila. And what I'm plotting here is that exact prediction I just showed. So this formula, but put into the heritability ratio. And I'm plotting it for two different strengths of selection. And instead of plotting it, I'm plotting it as a function of the number of loci that are contributing to the trait. So you can sort of think of that as like the mutational target size. But instead of measuring L as like a number of loci, I'm measuring it as a fraction of the total Drosophila genome, right? The euchromatic genome, at least. So this is like the percentage of base pairs that we need to contribute to the trait to get you the heritability shown on the vertical axis, right? The shaded region here is where we want to be, so something like 10 to 60% for our heritability. And you can see that, especially if selection is on the stronger side, we, at a bare minimum, to even get into this region, we're talking about 1% of all the base pairs in Drosophila chromatin needing to contribute to each trait, right? So for any trait to reach that point in this model, we need 1% of every base pair contributing. And if you want to get up to the sort of the 50% mark, you're going to be closer to about 10% of all base pairs in euchromatin contributing. And so there are, as Nick has showed nicely, like examples where there's extreme polygenicity and there's traits that have these crazy genetic bases, but this becomes a bit hard to swallow when we need it for all traits, right? And so that left a bit of a conundrum. So, it's just really hard to imagine that mutation selection balance isn't very important for determining VG, but our simple mutation selection balance models seem to be pretty far off explaining what we're seeing in nature, right? And so the obvious reaction to that is that our models are too simple, and we need to make them a bit more complicated. And people have done a lot of work on this. I'm not going to get into that now. There's these excellent review papers. Well, that's actually a chapter of a book, the second reference there. If you're interested in this topic, I strongly recommend reading them. But I will say that in 2005, Nick used the word embarrassing to describe the state of our theoretical understanding of this topic, because our predictions were still so far off the observations that we see. If you have an issue with that, you can blame him for that. So today I'm going to focus on a particular assumption that I think is actually possibly the most problematic one in a lot of these models. And that is that we assume that the fitness function stays still in these models, right? We know that that's not true, right? So the environment is changing all the time. This is not necessarily implying some kind of a trend or like climate change or anything. It's just all the time, just slight differences in what the population is experiencing, right? So we know that this is something that should probably be made a bit more realistic. But as soon as you take a step down this path, you stub your toe. And the reason is because environmental change is kind of a pain to work with from a mathematical perspective. We don't have sort of a standard model for like what's a reasonable thing that we should expect populations are experiencing. And to make matters worse, the predictions of models that have environmental change in them tend to be very sensitive to the assumptions you made about environmental change. So you end up sort of assuming your predictions in a way. And so two sort of quite well-known studies on this topic particularly, they were studying heritability predictions. They focused on cyclical environments. And so representing something like a seasonal cycle or something like that. And the predictions of these models are like sort of famously sensitive to how you structure your cycle, right? So if you have a cycle with the right frequency and nice big amplitude, you can get very different predictions from if you have a different frequency or smaller amplitude and so on. And so it's just not that helpful to explain these patterns that seem very useful if you need very sort of specific conditions for that to work. So I came at this from sort of the other end of like, okay, well, let's focus on like the boring stuff, right? So we know that the environment is kind of like wandering around slowly at minimum, right? And as a matter of fact, this is already the baseline assumption used for phylogenetic inference. So there they assume typically like, well, one of the most popular models is an Ornstein-Uhlenbeck process for the trait optimum. So they're basically saying the optimum fluctuates kind of like a random walk, a Brownian motion random walk, but it has some constraints. It can't go too far, right? Now the time scale for these models is very different, but it seems reasonable to assume that the most populations experience some movement in the optimal value for their traits. It's not necessarily strongly directional at any particular point in time. It's kind of fluctuating. And over longer periods of time, it might move further away. And as long as we make our sort of change slow enough, it's probably going to be pretty widespread, right? And so this I saw someone put their hand up. I'm not sure if I should.
Yeah.
Maybe I'll carry on to the end then. Okay.
Yeah. People should be asking questions on Zoom.
Okay. 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 So so this model is also nice to add to the Ladder-Bolmer model because it doesn't make it much more complicated. I've just added one parameter, right? Which is the rate at which fluctuations accumulate in the trait optimum model. So the rate per unit time that variance accumulates in the trait optimum. All right, so let's just look at some simulations quickly to get a sense of what this does. So if you put, if you let the optimum move in an OU process like that, you get what you see here in the gray curve. So the black curves are sort of the classic Ladder-Bolmer. It's actually not strictly speaking the Ladder-Bolmer because I've allowed drift to be in there as well. That's why it's a little bit stochastic. But I wanted to sort of have drift in both to be fair. So you see here heritability as a function of time. It's remarkably higher. It also goes like it fluctuates like crazy. And you can see the basic thing that's going on. It's not very complicated. If you look at the allele frequencies in the population, so under the Ladder-Bolmer model, you've got all your alleles are kept at low frequencies by stabilizing selection. If the trait optimum can move, there's this chance that you can drag alleles to higher frequencies, and that creates a lot of genetic variation when that happens. And in some cases, it can even drag some alleles to fixation. Okay, so this is all sort of fairly intuitive. The hard part starts when you try to sort of pin down how much of an effect this is, how big do you need sigma squared to be to have an effect, and so on. And the math here gets surprisingly challenging. So Nick spoke a little bit about how the infinitesimal model is nice because you can ignore genetic details. In this model, a lot of the effects are driven by the genetic details, and so you kind of need your foot in both quantitative genetics and population genetics. And it's a little bit of a hellscape there. So to get a sense of like one of the major complexities of this model, I'll remind you of sort of Fisher's fundamental theorem, which tells you that the rate of adaptation is proportional to the genetic variation. And so when the optimum is running away like this, the population is chasing after it, right? So it's trying to keep up with it. And the rate at which it's doing that is proportional to VG, and VG is the thing you're trying to predict, right? And so you've got, and VG is also determined by the fluctuations that are created by chasing the optimum, right? So there's kind of this feedback between adaptation and fluctuations where it's hard to get to the quantity you're trying to predict because you sort of need to know that quantity to know what the fluctuations are doing in the first place. So for example, if you have a population with a low genetic variation, it's going to really struggle to track the optimum, and it's going to create these large deviations from where it needs to be, very strong selection coefficients, and very intense selection, potentially dragging alleles to high frequencies. On the other hand, if it has high genetic variation, then it can track the optimum closely, right? So the thing you want is intimately coupled to the knowledge you need to be able to predict the thing you want to begin with. And so how much time do I have? I'll briefly try and outline some of the math that I got into to get a bit of an estimate of the sort of intensity of environmental change that's needed to get reasonable heritability values. But I think I'm going to have to skim through it pretty quickly. So you can see this is the standard selection equation that comes out of the Ladder-Bulmer model. So I'm not going to get into the details here. The key thing I just want to highlight is that this second term in this equation here is basically just scratched out when you have the trade optimum stationary, right? So you're going to focus all your attention on this guy here, and you're going to ignore the second term that behaves quite differently. And so it's in a way not surprising that you can get a qualitatively different model if you allow that delta to be non-zero. In particular, in this case, delta is not only non-zero, it's a stochastic process, right? And it's a stochastic process because it's the distance between the population mean and the trade optimum. The trade optimum is undergoing OU fluctuations. And you can actually show that the delta process also follows an OU process approximately with a restoring force that's proportional to VG. Again, the thing we're trying to predict. Now, that's a problem because OU processes are autocorrelated, which means that you can't just immediately use sort of any kind of diffusion methods or anything. So you need, ideally, a way to get rid of the autocorrelations. You can do that under some conditions, although it starts to break down within the regime of parameters that I was interested in, but it helps a lot. So you can sort of average over the autocorrelations and derive an effective diffusion. And you'll get an effective variance term that looks like this. So for those of you who are familiar with these models, you'll recognize like this thing here is the standard drift variance. And then there's this other thing here, which is the effect of the optimum fluctuations. And again, it's a function of VG, right? So that's a problem still. We haven't gotten VG out of the equations yet. To do that, we need to sort of do a self-consistency type thing. So the diffusion model predicts VG, and therefore, if you require that the VG that it used is consistent with the VG that it predicts, you can write down an equation for self-consistent VG. And if you do that and you make a few more other approximations that I won't get into, you can get quite a nice equation that looks like this. So this is the same equation we saw before in the first part here. So that's just the Ladder-Bulmer model. But you've got this additional term, which looks kind of strange. So it's got like a square root, and it's completely independent of the mutational influx. And so that's kind of cool. Like, it's telling you that, well, let me back up a little bit. So why is it completely independent of the mutational influx? So that's tied to this idea that there's a feedback, right? So the population kind of needs to track the optimum closely, otherwise it's going to experience more intense fluctuations, which will cause its VG to go up, which allows us to track the optimum more closely. That kind of chasing dynamic is sort of independent of the number of mutations that are segregating. It's just tied to the chasing dynamic. So it's not that surprising in a way that there's no mutational influx in there. But more importantly, for like predicting heritability, this means that if this guy becomes really small, which was the problem we had with the Ladder-Bulmer predictions, they were too small, the other term can in fact dominate, right? And your whole VG prediction actually becomes this thing. And because there's a square root in there, even if sigma squared is quite small, that number can actually be fairly sizable. And so I'll just briefly show that. I think we're running out of time here. So this is looking at the heritability predictions as a function of sigma squared. And you can see that even taking sigma squared down to sort of 10 to the minus 4, it's a very different model from the classic ladder-Bellman model, right? Even these really small fluctuations can have a really pronounced effect. And in a case like this, where you've got your ladder-Bellman model is hopeless, if you put in enough fluctuations, you can push your predictions nicely into the range of values that you'd like to see. Okay, I'm not really going to talk too much about the implications for heritability. I guess I'd just like to highlight a little bit the modeling interest here. So it's kind of an interesting case, right? Because there's this model that's very famous, very widely used. And if you just change one assumption and even make the parameter value a little bit non-zero, it like kind of sort of breaks the model completely. It becomes a qualitatively different thing. So I thought that was kind of interesting to share. And then separately from that, if anyone has any ideas for helping me with some of the math side of things, I would love to hear your thoughts. There's all sorts of issues with like averaging over the temporal correlations, like steady state is kind of meaningless in a model that fluctuates like this. I haven't even tried to get into the coupled dynamics of multiple variants under selection at the same time. So please come and bug me if you have a brilliant idea. Thank you.